Hello friends, I am Pandam Pawar, Assistant Professor and Head Department of Microbiology, Sri Venkatesh Arts, Commerce and Science College, Divulgaon. In this lecture, we learn about one of the metabolic pathway, and the key pathway in the metabolism is glycolysis. Everyone know about glycolysis. Glycolysis is a lysis of the glucose. Glyco stands for the glucose, and lysis is the breakdown of the splitting of the glucose and to produce the energy. It is one of the important metabolic pathways in metabolic processes to yield or to produce the more and more in ATP energy. ATP for the energy purpose. The ATP generation is initiated through glycolysis. And glycolysis is the uh, performed in the cytoplasm, and glucose is split by activating the glucose molecule by input into the ATP input, and is of initial stage is known as a preparatory stage, and second stage is a payoff stage. In a preparatory stage, the glucose is phosphorylated with the uh, phosphorylated by using ATP as energy, and in the presence of the enzyme hexokinase or glucokinase, is also known as a glucokinase, and the 1 ATP is utilized in the first stage of the glycolysis part. In second stage, the glycolysis or phosphorylated glucose 6-phosphate is again rephosphorylated with uh, sorry, the glucose is uh, again is isomerized and converted into the fructose 6-phosphate, and fructose in the presence of the enzyme phosphohexokinase hexoisomerase, or it's also known as a phosphoglucose isomerase, and uh, then a fructose 6 phosphate again phosphorylated with the 8 phosphorylated, and it convert into the fructose 1,6 biphosphate diphosphate, or is also known as a fructose 1,6 base phosphate, and in this stage also utilize a 1 ATP molecule, total 2 ATP molecule input in a preparatory stage. In first stage is glucose to glucose 6-phosphate conversion, and second is glucose 6-phosphate to the fructose 1,6 diphosphate, and in this step is followed by the enzyme, the phosphofructokinase, and phosphofructokinase is one of the important enzyme in the glycolytic pathway, or is also known as feedback inhibition, is also uh, introduced in these steps because of the allostatic enzyme. The phosphofructokinase is important role in the preparatory stage of the glycolysis, and then is fructose 1,6 phosphate is then splitting. Actual splitting is start in the next step is in the presence of the aldolase enzymes, and aldolase enzymes is converted into the uh, two hydrox is converted into the hydroxy, then the two molecules produced in the next step in the 3 carbon compound. The phosphofructokinase is fructose 6 phosphate is uh, 6 carbon compound, and then it's converted into the 2 3 carbon compounds in the next steps of the uh, glycolysis, and the next step is also known as payoff stage, because of the in this stage the more and more biomolecules or the energy is produced in the next steps of the glycolysis, and then is ATP input is payoff to the cell, and in is glycolysis is a preparative stage is important. All the compounds are the six carbon compounds, and the total three enzymes are used. In the first stage is a hexokinase. Second stage is phosphohexoisomerase, and third stage is known enzyme is a phosphofructokinase. Here total three enzymes. In this the is phosphofructokinase is important enzyme, and that is also known as allosteric enzyme, and this is responsible to regulate the glycolysis or the metabolic action in the cell. Because of the at the fulfillment of the ATP energy, this enzyme is inhibited, or this enzyme is feedbackly inhibited by the presence of the ATP. The more and more ATP, the inhibit the role of the phosphofructokinase, and due to this, the phosphofructokinase is regulatory enzymes is important as a for the enzyme cell economy and to the cell. And next is our stage. We learn about uh, payoff stage, and payoff stage is in a next stage the glucose fructose 1,6 diphosphate is split into the three carbon compounds. One is dihydroxyacetone phosphate, and second one is acetyl glyceraldehyde 3 phosphate. In the presence of the enzyme aldolase. Aldolase is the enzyme that split into the two compounds, three carbon compound. One is dihydroxyacetone phosphate, and second one is glyceraldehyde 3 phosphate. And these two enzymes is actually interconvertible into in the presence of the enzyme isomerases is are the isomers of each other, and hence is interconvertibly uh, in the cell. The next is glucose glyceraldehyde 3 phosphate is converted into the or again is phosphorylated with the inorganic phosphate in the presence of the in converted into the glucose glyceraldehyde phosphate converted into the 1,3 bis phosphoglycerate and inverse phosphoglycerate in the presence of the enzyme glyceraldehyde 3 phosphate dehydrogenase. In the presence of the enzyme glyceraldehyde 3 phosphate dehydrogenase, and this enzyme or dehydrogenase is inhibited by the enzyme uh, inhibited by the iodoacetate, and this can be steps is stopped due to the presence of the iodoacetate when introduced into the cell or the media. The iodoacetate then is energy producing steps or this stage is stopped or inhibited due to the inhibition of the enzyme of the glyceraldehyde 3 phosphate dehydrogenase enzyme. And next stage is 1,3 bis phosphoglycerate in then converted or the into the 3 phosphoglycerate in the presence of the enzyme phosphoglycerate kinase, and in this stage is the first stage of the to produce the ATP generation. Actual two molecules of the glycerol 1,3 bisophosphate is converted into the 3 phosphoglycerate, and in this stage is a total two molecules of the ATP yield, and this is a first stage at that the we yield a ATP in the glycolytic pathway, and then is 3 phosphoglycerate is converted into or this is isomerate into the 2 phosphoglycerate, and 2 phosphoglycerate is in the presence of the enzyme phosphoglycerate mutase, and phosphoglycerate mutase is converted into the 3 phosphoglycerate into the 2 phosphoglycerate, and next is a 2 phosphoglycerate is converted into the pyruvate sorry phosphoenol pyruvate in the presence of the enzyme enolase, and this enzyme is also inhibited by the fluoride when you introduce the media or fluoride, the fluoride inhibit the enzyme and stop the glycolytic pathway. And then phospho phosphoenol pyruvate is converted into the pyruvate kinase, and pyruvate in phosphoenol pyruvate converted into the pyruvate in the presence of the enzyme pyruvate kinase. Again, in this stage, we get two molecules of the ATP, two as in the form of the energy, and the second is a payoff stage to produce two molecules of the ATP. And next stage is a pyruvate is anaerobically uh, digested or anaerobically metabolized into the lactic acid, and the lactic acid that is accumulated in the cell when is anaerobic condition is present, then is converted into the lactate. Otherwise, the pyruvate is converted into the in the presence of the mm, in the presence of the pyruvate dehydrogenase is converted into the acid converted into acetyl CoA. In a pyruvate is anaerobically digested into the lactic acid, and during this stage is required the reduced energy in the form of the NADH, and NADH is required to recycle the electrons to convert the pyruvate into the lactic acid. And then is anaerobic condition, we get a lactic acid by the enzyme to the lactic acid dehydrogenase enzyme, and that is important role in anaerobic condition, anaerobic metabolic activities to produce energy when the oxygen level is very low in conditions. We know about everyone know about the when we are the anaerobic conditions in the such as the sprinters and others are the which accumulate in the muscles is the lactic acid muscle in the muscle, and due to the anaerobic condition or the due to the depleted condition in the oxygen. And next is pyruvate is also converted into the acetyl CoA in the presence of the pyruvate dehydrogenase complex or pyruvate dehydrogenase enzyme, and that convert into the pyruvate into the acetyl CoA to enter, and acetyl CoA then enter into the Krebs cycle, and it is important to the complete digestion or the complete metabolism of the glucose is must pyruvate entered into the glucose through the acetyl CoA in the Krebs cycle to acetyl CoA, and acetyl CoA is when entered into the Krebs cycle is totally reduced, totally oxidized in the form of the and to produce reduced energy in the glycolytic pathway, and then reduced energy is then is pumped into the and the Krebs cycle is totally run into the mitochondria, and mitochondrial and then this reduced energy is transported to the the ATP synthesis and then produced from the NAD from the reduced energy to produce more and more ATP to yield a maximum yield of the energy. In Krebs cycle is only run in the presence of the oxygen because of the to, to oxidize total oxidize the glucose to the required the more and more oxygen because of the it converted into a total conversion or total oxidation of the glucose into the CO2 plus H2, and in the totally conversion that we gain about 38 ATP in one cycle of the one glucose of the molecule. And the glycolytic pathway is very important in a metabolic action because of the we yield about uh, uh, 6 molecules of the ATP in the form of the reduced as well as the uh, two types of the ATP here we gain. One is ATP from the uh, in the form of the reduced energy. And again, the second is a substrate level phosphorylation, also in the two stages, is by the enzyme the pyruvate kinase and by the enzyme 3-phosphoglycerate. Here in these steps we get a 2 ATP, 2 ATP, total 4 ATP in reduced form, uh, substrate level phosphorylation, and the reduced energy also we gain in the form of the NADH2. And this reduced energy is then converted into, or then is transferred to the electron transport chain, and we get almost 2 ATPs, and the total is gain of the total gain of the glycolysis is a total 6 ATP in the glycolytic pathway. And when cell required more and more energy, then it transport or is glucose glucose is totally oxidized in the form of the CO2 and H2O in the cycle, in the Krebs cycle. And Krebs cycle is a very important, play an important role in the glycolytic, in energy production, as well as some biomolecules in the Krebs cycle is used as a precursor for the amino acid synthesis, as well as the some nucleotide synthesis. And here is important role in glycolysis, as well as glycolytic biomolecules, glycolytic pathway biomolecules also important role in produce or the synthesize and precursor for the amino acid synthesis. With this, the glycolytic pathway is totally completed. And thank you watching for this video.